The blue variety is one of the most common varieties we see in chickens. This variety is found in Silkies, Andalusians, Cochins, Polish, Orpingtons, Sumatras, and many more. Thanks to the blue gene, we also have the very popular splash variety, caused by homozygous blue. Knowledge of the blue gene is also very important when working with varieties such as lemon blue, silver blue, blue laced red, more. In this video, we will discuss the blue gene, how it works, and look over some examples of breeding blue to make blue, black, and splash, as well as a basic overview of Mauve and Platinum, which we will discuss in another full video in the future. There is a bit of confusion surrounding the blue variety, because even though the gene is called the blue gene, the blue variety only occurs when a bird is heterozygous for the blue gene. A bird homozygous for the blue gene is called splash. In this video, I will just be focusing on the blue gene specifically, but it is very important to recognize what makes up the blue we see in Andalusians, referred to as the Andalusian Blue variety or laced blue. The dark lacing we see in Andalusians is not caused by the blue gene. And keep in mind, this is not the same lacing you would see in a silver or gold laced bird. Although similar, there are additional genes involved in making the silver, gold, and chamois lace varieties, so I don't recommend crossing them with the Andalusian Blue variety. The blue as we see it in Silkies does not have these additional genes that make up the lacing in Andalusian Blue or laced blue. Keep in mind if you are raising Andalusians or other breeds that are laced blue, that you will need to pay attention to not only the shade of blue, but also the shape and size of the lacing, as well as other breed type characteristics. In short, one of the genes that makes up the Andalusian Blue variety is the blue gene, but not all blues, for example, Silkies, are Andalusian Blue. It isn't uncommon to see the term Andalusian Blue used to refer to the blue we see in the context of Silkies, even though the genes that make up the lacing aren't present in Silkies. The reason we go out of our way to call it Andalusian Blue in North America, even though that is incorrect in the context of Silkies, is because it is often confused with self blue, which in America is actually lavender, which is a totally different unrelated gene. From now on in this video, I will only be talking about the blue gene itself now that we have the specifics out of the way. The blue gene is incomplete dominant, meaning it looks different in heterozygous form than it does in homozygous form. I explain more about how incomplete dominance works in a video in our genetics series, which is linked in the description. The gene symbol for blue is a capital B, L, and the gene symbol for the absence of blue, which we will call black, is lowercase b, L with a plus sign. The plus sign is there to represent that the absence of blue, which is black, is the wild type, and the blue gene is a mutation. The blue gene is a eumelanin diluter, meaning it dilutes black and leaves phenylanin, gold, unaffected. Because the blue gene leaves phenylanin unaffected, it also leaves the phenylanin diluter silver unaffected as well. We can see an example of this in the variety lemon blue, which is made on the birchen base, gold, and has the blue gene. There is one more thing I'd like to cover before we move on to the different breeding combinations, and that is how to tell the difference between blue, black, and splash. It may seem more obvious on normal feathered birds like Andalusians, but it isn't always obvious in breeds like Silkies. Keep in mind that blue can come in a lot of different shades due to differences in gene expression, and it isn't uncommon to mix up blues and blacks. Thankfully, you can tell blues and blacks apart by sheen. Sheen is the green shine, sometimes purple, that is visible when a bird is standing in the sunlight. This should only be seen in black, not blues. It's very important to know your line and keep in mind where your line came from, because sometimes low-quality blacks actually don't have sheen. And sometimes very dark blues can appear to have sheen, but they don't, so you need to look closely. Splash is generally white with black and blue feathers throughout, or in Silkies sometimes looks more grayish with black and blue feathers throughout. Sometimes the blue and black streaks are more muddy looking and other times are well defined. The desired amount of these streaks is mostly up to interpretation, although the American Bantam Standard states that more than 50% blue in plumage is a disqualification. With all of that covered, let's go into the 6 different Punnett squares to show how to get the most out of your breeding program, as well as what is needed to make Mauve and Platinum at the end. The first pairing we will look at is black x black. This is a pretty easy one, as both parents are homozygous for BL+, the absence of the blue gene. All offspring from this pairing will be black. The reason I mention this is to show that you cannot get blue offspring unless one of the parents is blue or splash. The next pairing we will look at is blue x black. The genotype of a blue chicken at the blue locus is capital BL/BL+, and the genotype of a black at the blue locus is BL+/BL+. Since blue is an incomplete dominant gene, when you breed blue to black, 50% of the offspring should be blue, with a genotype of capital BL/lowercase bl+, and 50% should be black, meaning they have the genotype of BL+/BL+. Determining whether a chick is blue or black is something you can tell based on chick down, and if not, you can usually tell in a week or so once the feathers start to come in, on breeds with normal feathering. It might be harder to tell in Silkies, especially if yours are a darker blue. I find if you look in the primary feathers, even on developing chicks, if they're lighter, it's probably a blue. Although it isn't known why, when you breed a blue to a black, the resulting blue offspring typically come out darker. This isn't always the case, because there are definitely exceptions, but generally when breeders, myself included, are trying to get a darker shade of blue, you breed your blues to a black and get darker blue offspring, as well as black offspring. And when you are trying to get a lighter shade of blue, you can breed your blue chickens to a splash. Again, it isn't known exactly why this occurs, as it is up to gene expression, and a lot of variation can occur, but more often than not, breeding a blue to a black results in darker blues, and breeding a blue to a splash results in lighter blues. The third pairing is splash x black. Splash is the homozygous form of blue, meaning it has the genotype of capital BL/capital BL, and when bred to black, makes 100% blue offspring that have the genotype of capital BL/BL+. The main issue with this pairing in a breed like Andalusians is that it may be harder to select for a certain size of the lacing in Andalusian Blue, because you won't be able to see this lacing in black or splash birds. This isn't very important for breeds like Silkies though, and if you want to guarantee that you will produce blue offspring, this is the pairing to go with. The fourth pairing is blue x blue. Blue x blue is the best pairing to get the largest variety of colors in the offspring. Remember that the genotype of blue is one blue gene, capital BL, and one black gene, BL+. So when you breed two blues, 25% of the offspring should come out splash, with a genotype of capital BL/capital BL. 50% should come out blue, with a genotype of capital BL/lowercase bl+, and 25% should come out black, lowercase bl+/lowercase bl+. The fifth pairing is blue x splash. As mentioned earlier in the video, blue x splash is a good pairing to do if your blues are too dark and you need to lighten the shade. Blue has a genotype of capital BL/lowercase bl+, and splash has a genotype of capital BL/capital BL because it is homozygous for the blue gene. This pairing should result in 50% blue and 50% splash offspring. Our final pairing is splash x splash. Because splash is homozygous blue, meaning they have a genotype of capital BL/capital BL, breeding two splashes together will always result in splash offspring. The splash offspring from this pairing tend to be lighter, having less blue and black feathers from their parents. Again, this isn't always the case, but tends to be a trend with this pairing. And finally, with all of that covered, let's take a look at Mauve and Platinum. Mauve is the result of breeding blue or splash with sex-linked chocolate. If you aren't familiar with the genetics of sex linkage, I suggest watching my video on sex-linked genetics that will be linked in the description. Keep in mind that if you do use blue, mated to sex-linked chocolate, only 50% of the offspring will actually get the blue gene, and the others will be black or chocolate, depending on which pairing was chocolate. Again, chocolate is a sex-linked gene, so I recommend familiarizing yourself with the genetics of sex linkage before trying this. And finally, platinum comes from breeding blue or splash to a bird with the dun gene. Again, if the bird you are using is blue, only 50% of the offspring will get the blue gene. The same is true if the bird is only heterozygous for dun. Keep in mind that the dun gene is also incomplete dominant. The homozygous form of dun is known as khaki. In order to get platinum, you need to make a pairing that will ensure at least part of the offspring will get both the dun gene and the blue gene. The best way to do this, in my opinion, is to breed a bird that is khaki, homozygous for dun, and a bird that is splash, homozygous for blue. This will ensure that all offspring are platinum because all offspring will have one copy of the blue gene and one copy of the dun gene. All in all, we can thank the blue gene for many of the chicken varieties we enjoy today, like Andalusian Blue, Splash, Lemon Blue, Blue Laced Red, Mauve, Platinum, and more. As always, if you ever have any questions, feel free to contact me via any of the methods linked in the description. Thank you so much for watching, and I will see you in the next video.